We've got critical mass, and we'll begin now. With me, hopefully you can all see Luca's slides and face there is Luca Pinello, is assistant professor at Mass General and at Harvard Med School. Previously, he was an instructor and research fellow at the Dana-Farber Cancer Institute and has a PhD in mathematics and computer science from the University of Palermo. He's going to be speaking about one aspect of his lab, which is using innovative computational approaches and cutting-edge experimental assays for genome editing, certainly an area close to my heart, and single-cell sequencing to systematically analyze sources of genetic and epigenetic variation and gene expression variability in human traits and diseases. You might know him from some of the areas in recent news, including BCL11A enhancer dissection, chromatin state plasticity, and both the original CRISPResso and the paper, the updated version he'll be talking about today. With that, let me turn it over and we'll try and use a number of the question and answer features. So if you have a question as we progress, please type it in and we'll figure out if we can address it in the middle. I appreciate everyone remaining civil and keeping their names in here so that we can try and call on you possibly. There will be a few more of this VGETS series. James Dahmer and Samantha will be presenting from NIST. So keep an eye on the announcements as those roll forward. With that, let me turn the mic over. Dr. Pinello.
Thank you. Thank you for the nice introduction. And I think you can all hear me and see the screen, right? Probably yes. Okay. So yeah, it's a great pleasure to be here. And today I want to talk about CRISPResso2. That is a tool that we developed to analyze genome editing data. So So a quick disclosure, I'm a co-founder and shareholder of Cradick. So today I want to talk first about CRISPR genome editing really briefly and focus on the data generation. How do you get the data that we want to analyze? And then I will introduce CRISPResso2. I will give you a nice overview and all the features that are present in this tool. And finally, I want to conclude with a new feature that we are implementing in CRISPResso, essentially the prime editing quantification. And if we have time, we can discuss also other points that you want to know about CRISPResso for people that are already using it, like of targets analysis, for example. So let's jump in. So genome editing, if you're in this talk, probably you understand it's a really important area, and has changed totally our way to do medicine, biology, and biotech. And briefly, the key idea for people that are maybe not familiar with this is that you can modify a genome in a targeted way. So you can change a particular region in the genome and create insertion, substitution, or deletions. And with this, you can do many things. You can, for example, disable a gene, or you can, for example, investigate, the function of any gene of the genome with genome-wide screens and so on. So this is the key idea, and there are several applications of this. And regardless of which editor you use, so there are some common challenges. Why do we need to perform careful analysis of the data of genome editing experiments? So the first immediate question is did it work? So let's say you want to modify a genome, use a genome editor, and you want to know, did it work? Do I see the edits that I want to create? And another really important point, as I mentioned before, we want to modify the genome in a targeted way. So we might want to modify only a single region. So how can you be sure that you are not modifying other regions? So this is related to safety. Because, for example, you can modify a region that can lead to a disease or, or cancer. So you want to make sure that, you can quantify if, you are modifying the genome in the proper way. Another important thing is that, you can get, a number, but, sometimes it's better to visualize what you're doing to your genome. So what are the visualizations that we can use to effectively see if you are modifying the genome accurately. And for people that develop the technologies to modify the genome, it might be nice to have some tools to compare what different editors are doing to your regions of interest. So these are the three big picture questions that we're trying to solve when we go into the analysis. So genome editing, there are like different tools. I will just give you like a super quick intro so we can jump in the data analysis. So the most commonly used system is the CRISPR-Cas9 system. So just want to put the yeah, okay, I can use my mouse. So you have the CRISPR-Cas9 system in which you have the Cas9 protein, you have this guide RNA that allows to target this system anywhere in the genome, assuming that you have complementarity of the sequence and also a sequence that's called PAM, but these are details that we don't need for now. And after you deploy this system, it recognizes the sequence and will create a double-strand break. So the cell will try to repair this double-strand break. And there are like two common pathways. One that is called non-homologous end joining. They will create insertion and deletion. So the cell will try to repair this. It will create these insertion and deletions. Or you can provide what is called a repair template that will create a particular replacement. And this pathway is called homologous direct repair. So with this, you can modify the genome in a targeted way. However, this HDR is low efficiency. So it's not really easy to, for example, create substitutions. So recently, another class of editors has been introduced, the base editing. So the idea is that you have a modified version of Cas9 that doesn't cleave, doesn't create double-strand breaks, fuse with an enzyme, and this is called base editors. And like long story short, there are a lot of passages from steps from year to year, but the key outcome of base editors is that you can create substitutions in a targeted way. So this is like the key idea. So you have your cells, you modify, and you will create insertion or deletion or substitutions. So now we want to know if the experiment worked. So a common strategy is like to amplify the region that you're trying to modify. And you can design primers in this region, and this region will be called amplicon. And then after amplification, so you create multiple copies, you will sequence. And you can use, for example, Illumina sequencing. And after this step, you will get a FASTQ file that is a file that contains reads, essentially the sequences that you amplified. So now, like, how do you analyze the data, right? So up to here, you have successfully edited your cell, and you get your sequencing data. So you want to know, like, do I see any edits? Of course, it's not a file that you can open, in Excel or with Word and look read one by one, because you will have millions of reads, for large experiments. So you need a specialized tool to do this. So in 2016, we published this tool, the first iteration of CRISPResso, that try to solve this problem. So the basic steps, so you start from this FASTQ file that I was mentioning before, that contain all the reads that should cover the region that you're trying to modify. So the first step is that you want to align this to the expected amplicon that you amplify. So this is the wild type sequence. And then, given that you know which guide RNA you used to target this region, we can essentially create what we call quantification window. So here we can look specifically around these windows for the edits. And then, based on the reads that contain modification in this window, we can classify the reads in modified or unmodified. And finally, we create a summary of editing and several visualizations. So one thing that is really important, that sometimes create confusion, is like this quantification window. Why do we need this quantification window? The reason is because when you sequence, whatever technology you're using, there are like some sequencing errors. So you may see modification far from the expected cleavage site, and these are probably artifacts. So with the quantification window, you can reduce the false positive. So this is the key intuition of the quantification window. So now if we just look, at some initial visualization that we proposed back in 2016, the idea was to essentially first provide an overview of how many reads are unmodified. So these are reads that were non-modified or they were repaired perfectly. And then, in this particular experiment, we were trying to create homologous direct repair to trigger this pathway to introduce forced substitutions. But this is not always, the case. As I mentioned before, this is usually inefficient. So you can also trigger these other pathways simultaneously. So some reads, you will see, this different profile where you have, deletion. So just to orient yourself, what you have in the x-axis here is the reference amplicon position. So these are the different positions. And then, these three different curves correspond to insertion, deletion, substitution. And the dotted line here is the expected cleavage site. So that essentially compare, to this plot here, you start from this pie chart, and then, our tool will decompose this in different categories. And this mixed category that you see here, so these are artifacts that we are not sure how to classify. However, we improve a lot this visualization, as you will see soon. So this was intuitive, but you can have something much simpler to understand. Another thing that is really helpful when you try, for example, to do knockout. So you can also quantify frame shift or splice site. So for all the reads, assuming that you know what is the coding sequence, imagine that you are trying to target an exon of a gene. You can, after the convolution of the reads, you can quantify how many of my reads are creating, for example, frame shift mutation, if you want to disrupt the gene, or in frame. So this, in theory, may not be effective, but you can change amino acid. And then this other plot essentially tells you how many of your reads contain deletion that are modifying, a potential splice site. So this was more or less what we did in CRISPResso in the first version. Then we had also this little utility to compare two experiments. So this is really helpful, for example, if you do one experiment where you treat your cells with the guide of choice, and then maybe have a non-targeting guide as a control. So, and with this tool, you can create this kind of plots where you overlay the edits that you have in this amplicon where they're happening. And this is the other line is the control. So now if you take the difference, you see where, for example, the control was higher than the edit, and these are for sure false positive. So sometimes, this is a more or less clean experiment. Sometimes you see really weird things. For example, you may observe that you have some SNPs or other things that are you were not accounting for. So it's always a good idea to perform a controlled experiment. So this was more or less CRISPResso, and I was looking when this started, and this was a nice conversation we had with Daniel Bauer. So he started from a project that he started. So he was doing this PC11 screen. And then, we had this data, I had this data, we were really excited. So we created this pipeline for his paper. And, I was still a postdoc, this was a side project. And then, we realized together, maybe it's nice actually to share this with the community, because I'm sure many other people have, the same problem. And this was, in 2015. And also I was really naive. I was saying okay, this is the final pipeline, we did it. And in reality, things are more complex, and the CRISPR field has evolved a lot. So initially, we were focusing only on a single amplicon, as I just showed you, and then people started to look for multiple amplicons at the same time. For example, you want to measure your target site and maybe put putative of targets, or just to save money to pull multiple experiments. some folks started to look at whole genome sequencing data. And then, many more, based on what the community was doing, we started to introduce more and more features. And, the field has evolved a lot. And that's why when, I started my lab, Candace Kleiman was one of my first postdocs. And, it's a postdoc shared between my lab and the Jung lab. So we decided to improve CRISPResso, trying to take into account all the new things, the cool things that were happening in the genome editing field. So, and CRISPResso2 is Meili's work. So he turned, this simple tool into something, beautiful. So this is Candace Kleiman. And so the new feature, there are a lot of things that, introducing CRISPResso2. The first thing, was the inclusion of base editors, because, this is something really important that with CRISPResso1 was not really easy to analyze. And another thing is like the batch mode for analyzing and comparing multiple experiments. And another thing that we did was the allele-specific quantification. So this is an extension of, the HDR mode that we had in CRISPResso, but give you a much better flexibility in analyzing, for example, allele-specific edits. So if you want to target, for example, heterozygous SNPs. Another thing that we realized is that off-the-shelf aligners are not perfect, are not tailored for CRISPR genome editing. Outcomes sometimes you see some really bad artifacts. So we rewrote the aligner and also improved the speed dramatically. So CRISPResso2 is much, much faster. And this was published last year in Nature Biotech if you want to know all the details. But today we'll try to give you an overview to show like how can you use this tool. So the tools, in CRISPResso2, there are like different tools. So the first tool, so let me see, okay. Yeah, okay. So Kendall is on the line. So if there are like really technical questions, like you can, send to us and, he might be able to answer better than me. So we will have, maybe at some point, I will stop, I will take some questions. So if you have questions, you just write on the chat. So, and I will try to review. So yeah, I was saying, so these are some of the tools that we have here. So the first is like for simple amplicon deep sequencing. The second is like for the batch mode where you have like multiple conditions that you want to compare. And then you have like this pooled mode that essentially when you have multiple regions that you want to profile. And finally, we have this WGS that is for all genome sequencing data. All these tools share the same steps, more or less. So the first step is that you want to do some quality filtering for your data, because if the reads have low quality, you cannot trust the outcome. The second step is the adaptive trimming. So you may want to trim your reads for adapters or other low quality reads, low quality bases, and then we merge the reads. So often people use this parent mode. So you have like two reads, one from the left and one from the right, and usually we suggest that to have overlapping reads. This is like another thing I want to spend 1 minute because we receive a lot of emails regarding this point. So we suggest to have overlapping reads because you have more confidence in sequencing, for example, the location where you have like your edits twice, also to make sure that if your region somehow ambiguous, if you have two reads, you can anchor to the right location of the genome. However, you can use CRISPResso if you have single end reads, assuming that your expected edits are in around the middle of the reads. So, and finally, we have this alignment, essentially aligns all these reads to the reference, and after this you have the visualization. So these are a quick overview of the tools. Sorry, let me see, because I see there are already some questions. So maybe before jumping in the next section, I can let me share only the PowerPoint. Just 1 second. Okay. So
Yeah, you can go ahead and both Kendall and yourself jump in and answer some questions. I think there's a number of people that I saw some of them that it's hard to appreciate, you know, the importance of some of the parameters that you were discussing, such as the size of the window or the need to do these untreated controls, particularly if you're looking at very low events. So it might be a good time to address a few of those before you get to the improvements you made in CRISPResso2, where you address those more.
Yeah, sounds good. And so for the window, like, you know, the size that we suggest is like 1 base pair on each side. And when I say 1 base pair on each side, doesn't mean that, you know, you cannot quantify, for example, deletion or insertion longer than 2 base pair. So the way it works, when we say 2 base pair around the cleavage site, essentially anything that will overlap with this window will be quantified. So this is like essentially something that, you know, is really important to keep in mind, because sometimes we see people that use really large windows because they think like, you know, if I have to capture a large, you know, deletion, I have to enlarge the window. So there is no benefit in doing this, and you will capture more false positive. So the default setting essentially is like 1 base pair on each side, so a total length of 2. For base editors, you know, it's different, because, you know, like you are not cleaving the DNA, and, you know, like the window size, you know, is in that case is a bit larger. So, you know, there you may need to tweak depending on, you know, your application. But for Cas9, we just say, you know, keep at 1 base pair. So the second question, sorry, Kendall, do you want to add anything to this? I don't know if you can. If you're
I know, I just answered these in text.
Oh, okay. Yeah, actually, I cannot see the, I don't see the question, like trying to find the button, you know, they keep changing the interface, but maybe, you know, you can, you know, TJ, maybe you can read me the web, because right now I'm not able to actually open the question windows. So yeah, the other question was about the control. Oh, actually, I see the chat. Okay. Yeah, I cannot open the button.
The Q&A isn't going. Okay, one of the questions was on the level of detection, and some other questions, I think, that depend on the sequencing, you know, just exactly how big a deletion and such, which I think you can address.
Yes, so the level of detection, you know, is dictated based on your like sequencing, you know, strategy. So for Illumina, like there are a lot of papers that, you know, they claim is around 0.1%. So actually we did a simulation, and, you know, like sometimes we see much higher rates than 0.1%, but this was in a simulation. Real data, like the best way actually to estimate what is your sequencing error is like to use a control. So we'll use a non-targeting guide, you sequence the same region, anything that you will detect is a false positive. So this is like a quick way to have, you know, a sense of, you know, what is your background. But, you know, like I think, you know, around 0.1, I think is reasonable, something, you know, to keep in mind. So, okay, so let's, you know, keep going, you know, for now, and, you know, I'm happy to answer, you know, more questions. I want to show you like now, like the way you can use, you know, these tools. So Kendall did a fantastic job creating a super nice web interface. So you can actually just go online with your browser at CRISPResso2.pinellalab.org, and you will see this nice page, really clean, simple. And, you know, to use CRISPResso, essentially you just drag and drop your files, your reads that you get from the sequencing machine, enter the amplicon of the region that you wanted to probe, and the guide RNA. So in theory, the guide RNA like is optional, but we actually enforce to have the guide RNA because, you know, like we know like where is the cleavage site, and then based on this we can set the window. So it's always, you know, provide the guide RNA, and, you know, given that you design the experiment, you know that. for free. So, and after you enter all this information, you just click submit, and nice report will be generated. So one thing I wanted to highlight here, so if you see this section here on the left, so this is a super nice feature to get started. So if you have no idea about what to do, so you can click play, and it will populate this field for you. So you have a sense of what kind of data you need to upload. And we have different live tutorial for different application. So we have the classic one, homologous and joining, multiple alleles base editor, homology directed repair, and batch mode. And it's really simple. You just click play, and then you will see the report. And with the high here, you instead see the report directly. So let's take a look at the report. So the first thing that you get, when you get the report is this first, some information about the alignment and the quality of your reads. So these are the way you put in, and after you do quality filtering and how many reads are aligned. And then after this, you get a quick overview of what happened to your reads. So in this case, 72% of your reads contain a modification around the cleavage site. So this was a successful experiment. So this is also a way, for example, if you profile an off-target site, you want to validate an off-target site, yeah, you want to see like 100% or close to 100%, because you don't want to have any modifications. So another nice feature is this nucleotide composition plot. If you go to the website, actually it's a live plot, so you can over in the amplicon, and we'll create this nice window here. So yeah, you can see like for each nucleotide, what happened to the nucleotide. So in this case, if the bar is full, you're not changing compared to the wild type. In this case, there are a lot of things going on. You have like some insertion, and they have the legend here. You have some insertion, the black are the deletion. You see the stereotypical pattern of Cas9, and also you will see, like, for example, for base editor, the substitutions. So then here you have like something similar to what we had in CRISPResso. So here essentially you want to have a profile for each of the events separately. So insertion, deletion, substitution, and you see mainly at this site you have deletions. And here you see the quantification window is like 1 base pair on each side. And finally, we have this nice plot. I think this is my favorite, because this tells you everything about like your site. So here what we do, we just show you the alleles and the frequency. So this is the reference, and this is like in this case 26% were unmodified, and then here you have this deletion, you have a +1 insertion, and so on. So you have a sense immediately about all the information of your editing experiment. So before jumping the base editor, let's see if there are other questions for this part. I cannot, TJ, unfortunately, I cannot see the window. So maybe if you can read me if there are other questions, I will try to answer.
Just trying to see if there's a few new ones.
Okay, yeah, maybe I will pick
So start from the bottom, work your way up. We've got Connor Schida asking, for manually setting the cleavage position for staggered cut nucleases, do you use the TS or NTS, and does it make a difference?
So this again, is there?
For manually setting the cleavage position for staggered cut nucleases, do you use TS or NTS, and does it make a difference?
So for staggered, we don't have, like you don't, the way it works is like, the window will not care about if you're staggered, not staggered, we just quantify anything that you have, like within the window. So for example, for Cas12a or CPF1, so even if it's staggered, we don't do anything special. We just say like, this is the expected, we just take the middle of the staggered cut. So that's what we do in that case. But yeah, maybe there is a way to like, do differently, if there are like some ideas, but we don't treat this differently actually.
So I guess another way of phrasing that question, looking at zinc fingers, which have a 4 base, or the newer Cas systems, is there less dependence on precisely narrowing the window as there is with the 1 base that you kind of see with the Cas9?
Yeah. I think, like the way to accomplish this at the end of the day is like, enlarging the windows. So we have actually a custom mode. If you go to the website, so there's this custom. In this custom mode, actually, you can tune in, the window depending on your application. So you can, for example, enlarge. You can say, it's not clear that you have a single cut. It's not a clear single nucleotide. So you may want to enlarge in these cases for this other, nuclease. So yeah, that's the way I will do. In that case, we don't provide a, preset. Because there are like too many options, but that's the place where you can actually override the logic of our tool. Okay, so for base editor, right? So up to here, I just presented how can you analyze Cas9 data. So like I think it was 2 years ago, we realized that CRISPResso actually was not so great, to analyze base editors. So we realized like, maybe we should, touch base with the WU lab, they invented base editors. So they know like what is the best way to quantify these events. And, we started this really nice collaboration with Ollie Rees. And so Ollie really helped us to, create this report I will show you. And we try to like have some visualization that are really tailored for these editors. So just show you like one example. So these are for the different editors that they were playing with. So and this is also a nice example for this batch mode. So the batch mode, essentially you have the same reference sequence, the same amplicon, and each row here is a different editor. So immediately you can appreciate what each editor is doing. So untreated is your control and there is nothing. Cas9, you clearly see mainly deletions and insertion. And these are the different editors that they've developed over the years. And another thing that we do is like to highlight, for example, if you have a editor that's trying to get C to T conversion, we can just highlight these events and you can immediately see, the effect of the editor on this one. For example, this one was modifying mainly only this C, and this one was essentially modifying efficiently both. And of course, you can use this to do other things. For example, for their case, they were caring about product purity, so you want only C to T. So you can quickly compare the editors, and we have also specialized plots to do this, where you have all the substitution frequency in this quantification window, and you can see like which one preferentially have C to T. So another thing that we did, and there are a lot of other plots for base editors, so you can look online, but these are the main one. Another thing that I wanted to highlight is a little specific analysis. So the idea here is that sometimes you may want to disrupt only one of the 12 leads. And you can exploit this, for example, designing guides that are specific for a heterozygous SNP. So in this paper, for example, they were trying to disrupt this allele that is responsible for retinitis pigmentosa. So they use this guide RNA with a special PAM. And then they were trying to disrupt this allele. So if you run CRISPResso on this experiment, you will get a single table like this. However, this is not really helpful because now you're mixing the reference, the wild type and the P23H allele. So it's not clear, what's going on here. So what we did, we spent a lot of time trying to do this properly. And with CRISPResso2 actually get something like this. So you have like the modification that are specific for the wild type allele and the one for the P23H allele. So here the idea is that in theory, you don't want to modify this because this should be specific to the, this pathogenic allele. However, this can happen, right? So, and you get, in this nice pie chart, you can immediately see how well this worked. So essentially you want to maximize, this part of the pie chart. So you want to destroy the pathogenic allele and you want to minimize modifying the wild type allele. And, we spend a lot of time, doing this properly. And, we believe, we have a nice way to quantify this. However, there are still some cases where you cannot distinguish which one you're modifying. And the reason for that, if you have like deletion that delete your, SNP that allow to distinguish this, there is no way to do this. So there are also this, part of the pie chart that will tell you like, we cannot do anything with these reads. So this is like the basic idea of the allele specific analysis. So you can see immediately, you can also use this for HDR or more complex scenarios. So another really important part, people started to analyze more and more data. And so speed was really like a bottleneck of CRISPResso2. So we realized that, maybe we should rewrite this. And, like Kendall did a fantastic job. And, you can see like CRISPResso2 versus CRISPResso2 for different number of reads, like from, like maybe 1 day to, minutes. So this is much faster. But one thing that we realized rewriting this is that actually the alignment that we were using, there was, a tool from the Emboss suite called Needle, had some problem with Cas9. And, the main idea here is that, so when you try to align, the sequences, it's kind of, you're playing this game. So let's say you want to align these 2 sequences, you're trying to put this sequence below this sequence and somehow find the matches. And you can do this with different operations: match, mismatch, gap open, and gap extend. And each operation will have a score. And you want to find the best possible way to put this sequence below this sequence using these operations. And, the problem is like, sometimes actually you can do this in multiple ways that have exactly the same score. So then, you have a problem. You can say which one is correct. And, like if you have a general program that doesn't understand, what CRISPR is doing, it would pick one of these randomly. And, this is a problem because it can give you a really bad quantification. And just to give you an example, edge case. So this is one example that we saw, and you can go from 7.7% to 36%. And we were like horrified by this and said, wow, I mean, this is like a huge deal. And I mean, this is an edge case, but things can happen. If you actually see what's going on here, like, for example, this +1, insertion, CRISPResso2 is assigned correctly at the right location. But in CRISPResso2 was assigned, like outside the quantification window, and the same for these other, events. So, bottom line is like, you really need to, when you do the alignment, to account for these events. Also, we look other tools to see if they had the same problem, Cas Analyzer or CRISPR-Dove, and like in the same data, they also had incorrect alignment. So bottom line is like, either use CRISPResso2, or if you use this tool, you should be aware that, you may have some artifacts. So, okay, maybe I will, stop, 1 second to take questions, like now, before we jump in the last section, prime editing.
One just came up. If you still can't click there,
Yes.
what biological insights does the tool use to choose? And I think that's, maybe you mentioned that earlier, so you can rehash about the cut site, right?
Yes, essentially, yes. So we know like, that if you introduce a double strand break, often you will have, +1 or +2 insertion around the cleavage site. Also the deletion, they should overlap with the cleavage site. So that's essentially that we put some kind of priority, right? Based on what we know of the repair mechanism, we essentially encode these priority rules that will, push the aligner in cases where you have exactly the same score to preferentially select these solutions. So that's essentially the basic idea.
I can pipe in with one question while we take a look at the list.
Yes.
Or, in the one that you were showing where there's the repeat sections, this is
Yes.
probably the case where we see a lot of microhomology mediated repair. And that's probably the situations where you'd see a lot of this, right? And the kind of the examples where you'd see a dramatic difference with shifting the modifications into the window. And
Yes.
so is there some analysis done to look at MMEJ repairs versus ones that might be more canonical non-homologous end joining?
Yeah, that's a good question. I mean, in the current version, no, but like you're absolutely right. And like MMEJ is so predictable, right? So like some folks have developed tools, to predict, really well, these events. So I think, there might be some improvement, considering this. I mean, I think with the implementation that we have now, like we should not miss, any areas, but, your comment, is really on spot. Kendall, do you want to add anything to this point? Okay, so yeah, let's move to the next, to the final section, prime editing. I'm sure you have heard about, prime editing is a big deal. So it's an amazing technology that you can use to replace part of the genome without double strand break, and also from the WLU lab. So the basic idea, you have this system, there are like 3 components. So you have a Cas9 nick case, and you have, this PEG RNA or prime editing guide RNA, and then you have this reverse transcriptase. So the way it works is really beautiful. So you have, this system goes here, and you first, thanks to the PEG RNA, you find the location in the genome, then this will nick, one strand, and then, like the magic, will start. So the reverse transcriptase will use essentially what you have encoded in the PEG RNA to extend this region. And this is mediated by this prime binding site. So you can start from here. And then, this will essentially extend using the PEG RNA. And the trick, essentially, the edit that you want to put is encoded here. So now, like, you have a way to transfer from the PEG RNA the mutation that you want to introduce to what they call a flap. So you create this edited encoding flap. And then, like 2 things, can happen, because at this point you have this flap with your edit that you want to introduce, and then you have the wild type flap. And these things are in some sort of equilibrium. So now 2 things can happen. One, if the cell will decide to repair, excising this flap, you are back to the starting point. So you have the wild type sequence. So all this work is for nothing. However, if you excise the wild type flap, sometimes you get the edited sequence. And, especially if the repair, would use this strand to repair. So now they have a trick, and they call this P3. So you have another nicking cast. They will introduce another, nick here, and this will trigger the cell to use this template to repair, and you get, this nice outcome. So this is the basic idea. Like, you can read, all the details in the paper, but the basic idea, you have a way to introduce this, whatever you want, session deletion substitution without double strand breaks. So for us, that we developed CRISPResso, we realized, like, how can we, like, quantify, these outcomes? So the first outcome for sure that you want to quantify is, like, these are your wild type sequence. And if you jump all the way down to the expected edit product, you can simply say, I can just count how many times I see the wild type, how many times I see the prime edited sequences. However, there are other things going on in between, as I showed you before. So you have, this initial nick, you have, this other nick, and then, like, you have this reverse transcription and repair, that goes after the excision. So in reality, like in addition to just count the edited product, we should be really careful and look around, the first nicking site, because you can have, some indel here, the other nicking site, and together actually they can create a large deletion, and also around, the 5 prime, sorry, the 3 prime flap that you create. So around this region here, you may have also other events, for example, scaffold insertion. So these are the windows that we have. Like for Cas9, you have a single window. For base editor, you have another window that is larger. In this case, we need to introduce 3 different windows. So Candle, actually did already a first, plot of this, what, how can, this can be implemented, and, a first visual report. So that's where we are now. So we're really excited about this. So here, remember, you have these 3 quantification windows. So these are some data from the original Anzalone paper. And here you have like this bar chart where you have like unmodified reads, modified reads. And, in these categories, you have like the prime edit and modified, but you may have some edits here. So some additional, substitution, can be sequencing errors. And then you have also the scaffold. So if you look here, it's kind of really hard to see what happened. In this visualization, so actually it's much easier. So you have, the reference, and then you have the prime edit. And here they wanted to introduce this substitution. You clearly see that, this worked as expected. And then in scaffold incorporated, we try to collect, like other, events that are not, in the prime editing. For example, here you see that you may have deletions and some insertion that correspond to the scaffold. And, below you can see like the spacer guide RNA, and this is the extension, right? And here you can see here you have encoded the substitution. And here you have the other nicking guide RNA. So here you have these 3 quantification. I don't know if you see it because they're really faint, but this is the way, CRISPResso2, is working right now. And, soon we hope to have this online. So please, send us your feedback now, and, let us know what you think about this utility. And I want to conclude, my talk talking about the design, something that we are also super excited. So for prime editing, you design your peg RNA. So we had this problem. So Jonathan, that is a shared student between my lab and the Jan lab, decided to solve this problem, and created a fantastic tool called Prime Design. And this tool actually just appeared online today in bioRxiv, so you can read about this tool, and we are really excited. So this tool is a website. So we try to do things that are user friendly. So you can just go to primedesign.pinellolab.org, and to design your peg RNA, you just enter the sequence, and you can encode here your insertion, deletion, substitution with a really simple encoding, and immediately you get the recommended design. So you get here your peg RNA design and the other nicking design. And also, you can tweak this if you're not happy with what we propose. And you can change a lot of things here. Like the prime binding site, length, the reverse transcription template length, and the nicking distance, and so on. And you get this nice table with all the design if you want to really optimize your experiment. So another thing that John did, now that we have this tool, say, okay, why don't we look, why don't we essentially find all the design for CleanVar? That is a really nice database that collect also pathogenic variants. So he took all the pathogenic variants in this database and created designs for all of them. And the nice thing about this is you can actually target the majority of them, and you have 2 options when you use this tool that is called this database. You just click here and you can decide, do I want to install the pathogenic variant or do I want to correct the pathogenic variant? And I just enter the dbSNP ID or the CleanVar ID, and you get immediately the designs that you can use for that. Also, other things that we have in this tool is like a pool design, or you can use this for genome-wide screen or tiny screen. So it's really a nice tool, that we just put online. So check it out and let us know what you think. So, and finally, given this is almost 1 hour, I just want to wrap up, and then we can take questions. So in summary, CRISPResso2 is like an evolving tool that is trying to capture all the current way people are trying to edit the genome and trying to support this effort, providing accurate quantification and visualization of these editing events from next generation sequencing data. And to use CRISPResso, I show you the website. So you can just go CRISPResso2.pinellolab.org. But we have also like a command line version where you can tweak many more things. It's open source, so you can see exactly what we're doing internally. And you can also use Docker. So for example, if you have a Windows machine or some weird operating system, you can use Docker to run CRISPResso. It also is in Bioconda. And with this, I want to thank many people that made this possible. So Candace Clement really to turning CRISPResso1 in something amazing. Jonathan Su, like for prime design, and many feedback and suggestion for CRISPResso, all the lab members. Daniel Bauer for like supporting this idea since the beginning, and the Young Lab and also the W Lab for really providing us a lot of feedback and support during the development of these tools. And also my past mentors and collaborators for CRISPResso1, and last but not the least, the CRISPResso2 users. So thanks a lot for providing feedback, for telling us when the website is done, you need to analyze your experiment. So we really appreciate feedback. So please continue to do so, and also everyone, all my lab members and my funding sources. So thank you very much.
Thank you. I think we all appreciated your seminar today. I think that there were a few questions, and so we'll give people a few moments to type in some other ones so you can collate what you haven't addressed, and we probably go forward with that. So please take a moment and type things into the Q&A below, and we'll allow Kendall and Luca to take a quick look. I haven't seen too many jumping in at the last second, so it might be maybe this chance of taking a few Q&As along the line allowed people to jump in. We'll hear feedback whether that was appreciated. I thank you for that.
Yeah, maybe while we wait for questions, John, do you want to add anything for prime design that probably I didn't cover? Like anything I forgot to say that you want to highlight?
Hey, no, I think you touched on all the main points. I can definitely leave the link to our bioarchive preprint in the chat if others are interested as well. So I can do that now.
Yeah, thank you, John. Okay, does your read mapper affected by soft clipping? So in reality, we don't do any soft clipping because we don't start from a BAM file, so we actually realign the raw reads ourselves. So yeah, there is no soft clipping going on in our case. A good question.
A question that seemed to be re-answered or rephrased in a slightly different way was this question of using the controls when you looked at these sequences that you had some reading. Do you want to give some indication of how often you see this low level possible insertions, deletions, or changes in a untreated sample? And I think people might be surprised by how often you're at the edge of detection, and that comes into play.
Yeah, absolutely. So actually, one thing that we notice is that, if you look at all genome sequencing data today, I didn't have the time to talk about that, but, we see like actually a little bit more background noise in that case. I don't know why, but, this is like from the experience that I have. And, like typically like indel, are more rare than substitution. I think, the background rate for indels is much lower. I don't remember on top of my mind the exact number. But, like, I think, as I mentioned before, having the control, always having the control can really help you to, to get out of trouble. For example, if you want to quantify really rare off targets, if you don't have the control and you see something around 0.1%, 0.2%, like you don't know if it's true or not, right? So in those situations, it's really important to really have the control and, try to estimate the background rate. And you can use for doing this, this CRISPResso compare tool. So, a trick, for example, would be like if you have two controls and you compare the two, you can immediately see in the difference what is your, variability, right? Because in that case, whatever difference you see, it's just, an artifact. So that's another, trick, that you may want, if you want to be really careful to consider. Oh, thank you, Dimitri. Okay. I think if there are no other questions, like maybe, oh, okay, there's another one. So maybe this is a question for John, Nicholas. John, I don't know if you can read the question.
Let me look. I don't see it on my.
Yeah, maybe I can read.
Oh, I see it.
Okay.
Yeah, so that's a really good question, Nicholas. In terms of a recommendation, it's a bit hard right now to be very, very confident in terms of the RT length, but generally in the original paper, we kind of saw that a minimum of around 5 to 10 nucleotides of homology downstream of the edit was typically a safe bet. But any time that the original paper performed insertions or deletions longer than 10 to 20 nucleotides, they often had a RTT length after the edit of 34 nucleotides. So this has nothing to do with the total RTT length, the reverse transcription template length, but it's actually focused on the reverse transcription template length after the edit. I don't know if that was clear, but these are very general guidelines. I think as prime editing is better characterized in more comprehensive ways down the road, the design rules are going to be more and more clear. And these are things that we look forward to incorporating into the prime design website moving forward. But for now, those are very general guidelines that were things that we discussed and may not have been explicitly discussed in the original prime editing paper from David, but are just general recommendations for now.
Yeah, I think, you also answered the question from Wiley. Right, I think it is an excellent question. So bottom line, as John said, like, I think we still need to explore, this a little bit more. Okay, yeah, I mean, thanks to everyone, TJ and Wei, like, thanks a lot, for, the invitation. It was like really nice, and thanks for everyone that, joined today. And if you have any feedback, we're online on Twitter, just shoot us a message.
It was exciting to see your tweet with the link to the bioarchive today. So we'll take a look at that. I appreciate you getting it to us hot off the presses. Thanks to everyone. please share the word. We'll have more of these virtual gene editing talks. If you want to present, certainly message me. James Dahlman is slated for June 5th. A few other people are trying to come up with the time. I know there's been some questions about trying to make it so it's good for the coasts and the ships at sea and people over across. So let me know, and especially if you're hoping to present. Thanks again for Luca Pinella and team. I'm glad a couple of which were able to jump on. And thanks, and everyone take care and stay safe.
Thank you. Bye, everyone.
Bye.